Hello, I'm Kayesha and I'm a PhD student at the University of Leicester, and I've used UK Biobank data to help identify novel genetic signals associated with ACE inhibitor-induced cough. ACE inhibitors are primarily used for the treatment of hypertension, which affects 1 in 4 adults worldwide. They are largely well tolerated, but between 5 and 35% of users experience a dry cough, which often results in a switch to an angiotensin II receptor blocker, or ARB, which is in accordance with current clinical guidelines. This adverse reaction is poorly understood, and pharmacogenomic studies can provide insight into involved mechanisms and pathways. However, these studies are known to be limited by small sample size, as data on drug response and adverse drug reaction phenotypes are not widely collected at baseline. Therefore, I have harnessed electronic health records linked to UK Biobank and the EXCEED study to define a phenotype for ACE inhibitor-induced cough. My phenotyping algorithm identified cases who switched from ACE inhibitors to ARBs and controls who were continuous users of ACE inhibitors. And combined with imputed genomic data linked to both cohorts, I performed a stage 1 meta-analysis which, thanks to UK Biobank, had a sample size approximately 7 times that of the largest previous genome-wide study of this phenotype. A joint meta-analysis with Stage 2 study, the eMERGE network, identified 5 independent sentinel variants. Of these, 1 has been previously described and 4 are novel. Fine mapping of these loci were performed to identify putative causal variants for variant to gene mapping, which included 5 well-known elements. Among the mapped genes, as expected, KCNIT4 was identified, as well as 2 genes, NTSR1 and PrEP, which are both involved in the mediation of neuropeptide activity, which supports hypotheses which suggest this adverse reaction is a response to the accumulation of inflammatory neuropeptides upon ACE inhibition. Strong genetic correlation with chronic dry cough and further analysis of the NTSR locus in a PheWAS study identified genetic overlap with chronic cough phenotypes defined in UK Biobank. In summary, this study has identified genetic signals and putative causal genes for which functional follow-up has given insights to the currently unclear mechanism of ACE inhibitor-induced cough susceptibility. Identifying genes and pathways underlying drug responses provides a crucial first step towards identifying why adverse reactions occur and how to predict and prevent them. Thank you.